Hi and Assalamualaikum. We are from Group 5 and we will be presenting a topic about alcohol, driving under the influence. The analyte that we want to observe is ethanol. These are our group members: Nur Atira, Nur Amira, Shaza Iman, Siti Nofilzah, and Nurul Faizah. The content that we want to present is introduction, samples and chemical found in the incident, sample preparation method, sample analysis method, proposed sample preparation and analysis framework, and conclusion.
Driving under the influence (DUI) is defined as the act of driving, operating, or being in control of vehicle while under the influence of alcohol or drugs to the point where the driver is unable to safely operate a motor vehicle. In Georgia, United States, a 16-year-old girl died in a car crash driven by a 17-year-old boyfriend who lost control due to drug influences. The death has led to the boy being charged with DUI, and vehicular manslaughter was sentenced to 50 years in prison, including 9 to serve. The wrongful death suit declared that the store sold the driver had the alcohol despite his license stated that he was under 21, as well filed by the parents of the dead girl. In Georgia, however, drivers under 21 are charged with DUI if their BAC is 0.02%, and if chemical test indicates that they have a small amount of alcohol in their system, they could also be charged with DUI and minor accident. Driving under the influence of alcohol or drugs have caused a huge number of fatalities, which for decades have steadily declined in the US. However, the numbers have always been unacceptably high. It has been statistically proven that more than 10,000 Americans are killed by drunk drivers every year. Fatalities sharply increase, especially on holidays. US statistics and trends discovered that 22.5% of the drivers are 21 years old or older confess that they drive while intoxicated at least once in 2021, and 12% admitted that they have been doing it frequently. Connecting 28% of fatalities to legally intoxicated drivers represents an appreciable drop from 2010, when drunk drivers were responsible for 31% of auto fatalities, and sizable decrease from 1990, when impairment caused 40% of roadway deaths. Next chart shows that young grown up was addressed to be the most hazardous age group for drunk driving. However, that rate slowly tails off as age increases. Compared to females, male motorists are 1.5 times more likely to be legally drunk and involved in car crashes. Alcohol is the present drug that has the ability to slow down the activity of the central nervous system, including the brain, and it may lead to impaired vision, making reaction times, concentration, and vigilance a lot slower. Muscles that are responsible to control the eyes are relaxed by alcohol, causing the vision to be blurred and visual reaction time to become a lot slower. The penalties for drunk driving charges in the US involve both criminal and administrative. As anti-DUI policies have driven down violations and fatalities, arrests have also been declined. Between 2010 and 2019, DUI arrests has dropped 31%. Despite that, DUI still holds 10% of the nationwide arrests, which is twice the number made of all other violent crimes.
So the chemical found in this topic that our group revised is ethanol. So alcohol in drinks like beer, wine, whiskey, champagne, and many more types of alcoholic drinks are specified as ethanol, or also known as ethyl alcohol. So ethanol is a clear colorless liquid which is a byproduct of plant fermentation. It can also be found in grains, fruits, and vegetables. For instance, vodka is made of sugar that's contained in potatoes, while wine is made of sugar from grapes. So ethanol is the primary ingredient in the drinks that causes drunkenness. A study conducted by Costardi stated that alcohol is a depressant to the central nervous system. This causes the neuron in the brain to fire off nerve impulse more slowly. The blood alcohol concentration, BAC, rises, and that's when the feeling of drunkenness occurs due to alcohol being consumed faster than the liver can actually break it down. The liver can only break down a certain amount at a time, so when it is overly consumed, the liver cannot take it. This table here shows the types of alcoholic drink with its ABV, which is alcohol by volume. So what we can see here that vodka has the highest percentage of alcohol by volume. The figure shows the metabolic pathway of ethanol in humans' body. So the metabolism of alcohol results in the production of acetaldehyde and also acetate. Acetaldehyde has the ability to bind to proteins, including microtubules, enzyme, and also microsomal proteins. Also, it combines with DNA to generate carcinogenic DNA and the neurotransmitter dopamine to form salsolinol, which may contribute to alcohol dependence.
In the determination of alcohol in breath sample, the sample can be tested by using the breath alcohol analyzer, which is known as Oregon DUI Intoxyler 8000 breath testing. So there were no specific sample preparation method needed for the process. The driver only have to breathe out the air in their mouth directly to the device. So the concentrations of the alcohol must follow the standard sample, which is 35 microgram per 100 milliliter air. The device that were used to analyze the alcohol level were applying the usage of fused cell or infrared cell. Infrared radiation is directly passed through the sample using an infrared cell, and any unabsorbed energy on the opposite side is measured. As expiration continues, the concentrations of alcohol rises and the breath sample is analyzed once the ethanol level has stabilized. This enables precise alcohol readings and implies that the amount of air required depends on the size of the person's lungs. The accuracy of the sample testing was totally depends on the breath from the deep lung air. Alcohol can be isolated from blood samples using liquid-liquid extraction (LLE), which then the detection can be proceeded with mass spectrometry. During the sample pre-treatment, 20 ml of blood are taken and 6.25% of volume over volume of 300 ml of aqueous ammonium hydroxide in aqueous methanol are added to the sample and truly mixed. Noted that the volume is 10 liter and the flow rate is 0.25 milliliter per minute for the pre-treatment sample injection value into the HPLC-MS. The amount of ethanol in the sample was then compared to the internal calibration plot. The detection of alcohol in urine sample are the harmless and painful method. The sample were first collected 100 microliter into the 20 ml headspace vial, and the filled vial were then placed into the vial rack of an automated sampler and was operating in SSCC. The septum piercing needle of the SPME device was inserted through the septum, and the fiber was forced out of the needle and left exposed to the vial's headspace for a minute to allow the absorption of ethanol. The other method that can be used for the detection of alcohol in urine and blood sample is headspace solid phase microextraction (HSPME) with the usage of carboxyl polydimethylsiloxane coated fiber.
To detect ethanol in human body after taking alcohol, we found that there is three methods that can be analyzed. First, headspace gas chromatography with simultaneous flame ionization. Second, liquid chromatography tandem mass spectrometry. And lastly, breathalyzer. So the first one, it is a standard method developed by using the headspace gas chromatography (HSCC) coupled with flame ionization detector. This method is well known due to its advantages such as rapid, reliable, and cost-effective. This approach for blood alcohol concentration analysis is crucial because due to the high occurrence of ethanol in forensic and clinical casework. So for this method, the mobile phase used is gas, for example helium, and the oven temperature is set at 50 degrees Celsius and can run the analysis. So based on the GC principle, the blood alcohol content can be measured directly by headspace gas chromatography of a blood sample. However, this method is not recommended for thermally labeled sample. So for next one, liquid chromatography mass spectrometry, which is LCMS, it is a diagnostic analytical technique that may be used to analyze a wide variety of chemicals. So it is a unique, simple to run, other than verified as a means of validating recent alcohol use. So usually, in this method, we use urine as a sample, and the urine sample we obtain at various time intervals from 6 hours until 72 hours before we run the analysis.
And the mobile phase we use is liquid and it can be either polar or non-polar. So this method advantages are it is cheap and it can analyze mixture containing multiple components. However, in contrary, it is limited to types of sample and it provides low concentration sensitivity. So moving on to the next part, a breathalyzer is a device that uses a breath sample to determine the blood alcohol concentration. Breathalyzer components semiconductor do not directly detect the presence or concentration of alcohol in the blood, which involve analysis of a blood sample, but they estimate BAC indirectly by taking a reading of the amount of alcohol in a person's breath. To use the breathalyzer, the subject is held into a test chamber filled with a reddish-orange solution of potassium dichromate through the mouthpiece. The ethanol vapors in the breathalyzer initiates a chemical reaction with the molecules contained within, converting them from orange to green. So the greener the color indicates that the ethanol is high. However, the accuracy of this device is not significant, hence it mostly used by most police to support an arrest.
In Proper's method for sample preparation and analysis framework, first we collect the blood samples. Then we prepare a stock solution of ethanol and N-propanol in 100 ml purified water, then store in a 4 degrees Celsius refrigerator. And then we prepare standard solutions by diluting the stock solution with purified water and with different concentrations for ethanol and N-propanol. The sample extraction will be done by SSCC. The blood sample will be diluted with 1:5 ratio with deionized water. A blank blood sample is placed inside a headspace vial follows with blood sample containing ethanol and N-propanol. Then, when sodium dithionite and ammonium sulfate are added, in vial is heated to 60 degrees Celsius. Fiber is then exposed to the headspace for 15 minutes in vial. After that, cover the vial in the headspace for 15 minutes at 50 degrees Celsius. The sample analysis will be done by SSCC with the temperature at 50 degrees Celsius and 10 minutes duration time. Blood sample is used in our proposed method because blood sample is easy to get and it is the most accurate and recommended way of testing concentration of alcohol in a person's body rather than using urine or breath. Sample extraction is by using SSCC because the sample is used in liquid form, which is the blood. It is also a fast technique as it applies automated process and requires less chemicals. SSGC is used to analyze the sample because it is the usual method for ethanol analysis and due to its ease of automation, sensitivity, and accuracy.
To conclude, the best method recommended to measure the alcohol content of an individual is through headspace solid phase microextraction using blood sample and proceed with analysis by headspace gas chromatography with flame ionization detector. The reasons why blood is so much accurate compared to breath and urine tests are because SSCC is a very simple and also automated method that doesn't need much chemical or solvent. Therefore, this recommended method is most suitable way for authorities to detect and measure the alcohol content for people who drive under the influence. We hope if this method, driving under influence related cases will drop and everyone will play their roles to spread awareness of how big impacts driving under influence may have not only on the victim but also on their loved ones as well.